Our last speaker for this session is Sirohi Kumar. She's an incoming first year at Smith College, and she's interested in working in the intersection of hard science with law policy and ethics.
Hi everyone. Thank you, Sam, for the introduction. As Sam said, my name is Sirohi Kumar, and I was the other summer student in the Dumont Lab. And my project focused on profiling the transposable element landscape in a new wild mouse resource. So my project worked to address the lack of genetic diversity in available mouse models. As most of you know, in order to decrease the amount of variability in experiments, scientists use model organisms that are genetically identical or as close to genetically identical as possible. At JAX, a lot of that research is done in the C57 Black 6J mouse line. While using these identical mouse models allows us to be more confident in our conclusions, it also limits the applicability of our findings. A single mouse is not representative of the whole mouse population. It would be like trying to study a disease in humans, but your only model is an 18-year-old Indian girl from Maine. If you try to apply your findings to, say, a 50-year-old fisherman from Alaska, you might run into some problems. To address this lack of genetic diversity, Dr. Michael Nachman of UC Berkeley created the Nachman mouse strains. So these are mice that were collected from various climate regions across the Americas and inbred to serve as better and more representative models of the whole mouse genome. However, we know very little about these mice because they're so new. In my project specifically, I worked with the Saratoga Springs, New York, and the Manaus, Brazil strains. So my project explored transposable elements. Transposable elements, or TEs, are segments of the genome that are capable of self-replicating and mobilizing. There are also mechanisms for rapid adaptation. Their mobilization and insertion throughout the genome alters gene expression and promotes recombination at a much more rapid rate than single nucleotide mutations. This allows organisms to express new phenotypes that, if selected for, could increase population fitness. However, at the germline, TE insertions are largely neutral and even deleterious, meaning harmful. They can result in disease states or even lethality. TE insertions in somatic cells have been associated with conditions like cancer, neuropathy, and autism spectrum disorder. As such, most organisms have developed regulatory systems that silence transposable elements, rendering almost all transposable elements inert in mammals. My project focused on class 1 transposable elements, which make up large portions of mammalian genomes. As you can see in this bar chart, class 1 TEs make up anywhere from 1/2 to 1/3 of mammalian genomes. A quick note on nomenclature, because this bit gets really complicated. Class 1 TEs are a class, and then within class 1 TEs, there are different families. So those sections of the pie chart we saw earlier, so lines, signs, and LTRs. And then each family can have multiple subfamilies. So I'll get into that a bit later. In order to better understand the genome of this new mouse resource, in my project, I analyzed the differences in transposable element accumulation between the new Nachman and Black 6 strains. This was a computational project. I analyzed short read data from 14 Black 6 samples and 8 Nachman samples. I then used JAX's high-performance computing cluster to analyze the data using a tool called DNA Pipe TE, which is a non-reference-based transposable element identifier that utilizes raw reads to locate transposable elements in a genome while taking into account genome size and coverage. I then analyzed my results using RStudio. So the first question I asked was, at a very high level, are the number of transposable element families different? So at first glance, this is just the proportion of total annotated TEs that fall within each family. And we have the Black 6, they're the first 2 samples on the left, and the Nachman are the rest of the samples. And at first glance, these proportions look very similar. This is the same percentage of annotated transposable elements, but this time separated by family. So we see that our results do validate the literature. Lines have by far make up the most annotated transposable elements. LTRs and signs have very similar levels of accumulation, and DNA transposons make up the least. Using statistical analysis, I found that there was a significant difference in the line and LTR-TE families. Specifically, black 6 mice had significantly higher accumulation of lines, and both the man and the SAR samples had significantly higher accumulation of LTRs than the B6. So the next question I asked was looked at a bit more detail. Are there specific differences in the accumulation of transposable element subfamilies? So in this plot, I have the percentage of the total genome that each subfamily makes up, where each subfamily is a point, and they're color-coded by the family that they fall within. So on the x-axis, we have the percentage of the B6 genome, and on the y-axis, we have the percentage of the Nachman genome. So for instance, families in the top right corner have high accumulation in both the B6 genome and the Nachman genome. There's a very high correlation here, so you can see by this line, which indicates that at first glance, it looks like there's a strong linear relationship, which means that the percentage of each genome that each subfamily takes up is very similar. However, if you look closely, you can see that there's some serious clustering along the x and the y axes. These were very interesting to me because the point of my project was to analyze differences between the Nachman and the B6 samples. So samples clustering at the x and y axes, or subfamilies clustering at the x and y axes, would be subfamilies that are unique to either the Nachman or the B6 genome. So my next and last question was which subfamilies are driving the differences between these samples? So I used a principal components analysis to answer this question, or a PCA. A PCA takes high dimensional data and then projects it onto a 2D space and allows us to see the differences between these samples. So which factors are associated with the most variation. So this PCA shows that there's very strong clustering of the black 6 samples, and the Nachman samples tend to separate along principal component 1 on the left. And if you look closely, you can also see that there seems to be clustering by sex, with mostly male samples clustering towards the bottom and mostly female samples clustering towards the top. So those are separated along principal component 2. When we look at the loadings or the individual transposable element subfamilies that are associated with most of this variation, we can see that the separation along PC1 of SignRep MM and GSAT MM are subfamilies those are satellites subfamilies, and along the separation along PC2 of these two subfamilies are line ones, which we were really interested in. So these subfamilies are part of the line 1. These are part of the line 1 subfamily, which is documented as one of the most rapidly evolving and actually recently active transposable elements in black 6. You can see our subfamilies of interest are in the top right corner, which means they're some of the most recently documented. This is really interesting to us because, as I mentioned at the beginning of this talk, transposable elements, particularly line ones, are associated with conditions like cancer, neuropathy, and autism spectrum disorder. So, but the biomedical research done associating these transposable elements with these diseases have been done only in black 6 mice. So the fact that there's a significantly lower number of lines accumulated in our Nachman samples, which again are much more representative of the whole mouse genome, indicates that these strains have unexplored variability that could make them very interesting models for these diseases. So in conclusion, I found that the general composition of transposable elements seems similar, but there are very key significant differences, especially between transposable element subfamilies that have been documented as recently active, and found that the variability in these Nachman strains as transposable elements will make them very interesting models for specific diseases. And just a quick note on future directions. I used short read data for my project, but we had long read data available for many of these Nachman samples. Long read data allows us to make more accurate and confident identification of transposable elements. It's a more accurate and confident as an identification of transposable elements. So we were really interested in using this tool called REPET, which is a long read specific transposable element identifier, and then comparing annotations to those found by a short read tool. So that's the next step for this project. Lastly, I would just like to thank my PI, Dr. Beth Dumont, and my mentors, Alexis and Laura, for helping me throughout the summer, and the SSP staff for making it possible for me to work here. Thank you.
Great work, Sirohi.
Thank you.
Why do you think there's so much variation within the Black 6 strain?
That is a good question that we anticipated that we would get. I will say in regards to this, I'll answer the question in two parts. In regards to this PCA, because the way a PCA works, small variations between samples could lead to very large sort of changes in this case, so that could explain some of the distribution this way. In terms of this box plot, which shows the variation in the Black 6 samples, quite honestly, we're not sure. This would be an area for further exploration because one would assume that genetically identical samples would be very genetically identical.
Do you think alignment of short reads might be the issue here?
It's definitely a possibility, I think, and using the long read data would help us validate that. Yeah.
What's your biggest challenge working on this?
Thanks for asking, Claire. As I mentioned, in terms of future directions, I actually started out this project. Trying to analyze long-read data using this RepEt tool, but it turns out there's a technological incompatibility between the tool and the JAX's high-performance computing cluster, which was totally out of my hands. That meant that we spent about 2 weeks trying to make two things work together that literally couldn't. So that was a frustrating experience, but also illuminating, I guess. So that was one of the big challenges. Any other questions? All right, thank you very much.